Today we are going to discuss about then respiration part 4. Today's discussion we are going to discuss about electron transport system as well as respiratory quotients. Good morning my dear students. In this session we are going to understand that is electron transport system. It is the last step of aerobic respiration in the electron transport system. That complex form of energy which was formed in glycolysis, Krebs cycle and link reactions converts into simple form of energy. NADH2 form of energy converts into ATP form of energy, and FADH2 form of energy also converts into ATP form of energy. Means complex form of energy should be converts into simple form of energy, that is ATP. This reaction takes place, this changes is possible through electron transport system. Electron transport system takes place in inner membrane of mitochondria, especially on elementary particle, that is in crystal. In this, the importance of electron transport system is that point 1 is oxidation of NADH plus H plus or NADH2 and FADH2 through electron transport system is means they converts into simple form of energy. The metabolic path through which electron passes from one carrier to another is called electron transport system. It is the definition of electron transport system. Anyhow, electrons have been transporting from one electron acceptor to one more electron acceptor as like a chain system. In this process, ultimate result is ATP formation. That is an electron transport system general concept. The third point is the electron transport consists of a series of coenzymes and cytochrome that takes part in passage of electrons to its ultimate acceptor, namely oxygen. Means these electrons are passing through electron acceptor. Those electrons are finally accepted by oxygen molecules. Means in the respiration process, oxygen is an important component, but oxygen is used at the end of this respiration process, that is electron transport. Then oxygen acts as a final hydrogen acceptor. Means oxygen is acting as a electron acceptor, oxygen is acting as a hydrogen acceptor. Hydrogen plus electrons together is accepted by only oxygen. All together is formed as a water molecule. So then water molecules are formed at the end of this electron transport system. The utilization of oxygen takes place in this electron transport system. Means in the Krebs cycle and glycolysis, oxygen is not at all used. Then fifth point is electron transport system takes place at inner mitochondrial membrane. Just now we discussed glycolysis takes place in the cytoplasm of a cell. The link reaction takes place cytoplasm of a cell with the mitochondrial in between. The Krebs cycle takes place in matrix of mitochondria, but electron transport system takes place in inner membrane of mitochondria, especially on crystal elementary particles. We can see few steps in the electron transport system, otherwise called as chemiosmosis. The first step is NADH gives rise to two electrons to NADH dehydrogenase. It's the first step. The second step is electrons pass through the electron transport chain, then releasing energy. Energy is used to pump protons across the membrane into the inner membrane space. Build up hydrogen ions, means protons constant ions in the space creates electrochemical gradient. These steps takes place in between mitochondrial matrix and the space in between two membranes of mitochondria. Means that reduction of NADH2 by reduction process, NADH2 gives rise to two hydrogen ions are separated from NAD. Then hydrogen ions are going to split as protons and electrons. Electrons have been moving through electron acceptors, that is electron transport chain. That the protons are moving from one side to another side, that is from matrix into inner membrane space. From time to time, the protons concentration have been increased in inner membrane space. At last, the high concentrated or high gradient protons are moving from inner membrane space into the matrix through ATPase enzyme. At the time, ATP synthesis is possible. These are more 2 points. The proton ions wants to diffuse back across the membrane but are unable to and must use special proteins to channel called ATPase. Means the protons want to come back to the matrix of mitochondria but is not possible to come through the membrane. It can be coming outside through special pores, that is a protein channels called as ATPase. It is also called as a complex 5. The pores in that protein channels contains ATP synthesizing enzyme called as an ATP synthase. So as an proton ions moves through the channel, that is nothing but ADP plus inorganic phosphate together is formed as an ATP. ATP generation is possible in the electron transport with the help of proteins proton concentration gradient. As usual, chemiosmosis process which we discussed in the photosynthesis process. Dear students, now we can try to understand the process of electron transport system. There are 5 complexes are present in electron transport system. They are called as in complex 1, complex 2, complex 3, complex 4 and complex 5. What is an complex 1 consist? Complex 1 consists of NADH dehydrogenase enzyme. It contains FMN flavine mononucleotide. This FMN consists of iron sulfur. It is as a protein. This complex is responsible for passing electrons from mitochondrial NADH to ubiquinone reductase. Means first electron acceptor is complex 1. That electrons are coming from NADH2 by reduction process. Complex 2, it consists of a enzyme succinate dehydrogenase. It also consists of flavoprotein or FAD, flavine adenine dinucleotide. This FAD consists of iron and sulfur protein. It receives electrons from succinic acid and pass them to ubiquinone. This process is called as an oxidation of NADH2. Means oxidation of NADH2 takes place here. Means NADH2 going to lose H plus, it's became NAD. Ubiquinone, it is a lipid soluble mobile carrier between complex 1 and complex 3 and also present in between complex 2 and complex 3. It is electron carrier in between complexes. Complex 3, it consists of cytochrome C reductase enzyme. It consists of electron carriers, namely cytochrome B and cytochrome C1. It contains iron sulfur proteins. It participates oxidation of NADH2 as well as FADH2. Main NADH2 oxidation takes place only in the complex 2 and complex 1, but FADH2 oxidation takes place in the complex 3 only. Means by oxidation process, NADH2 going to produce protons, FADH2 going to produce protons. Based on the proton concentration or gradient, ATP synthesis will be depends. The third one is an complex 4. It consists of cytochrome C, oxidase complex. This cytochrome contains cytochrome A and cytochrome A3. This cytochrome A have that copper ions. Then they receives electrons from cytochrome C and pass them into that oxygen. Then 2 protons are needed to form that water molecule is formed. Here at end of the electron transport, water molecules are formed along with that ATP formation of energy. But these water molecules are formed with the help of protons released from NADH2 as well as FADH2. Electrons are released from FADH2 and NADH2 along with its one oxygen participation. Means in the aerobic respiration, Oxygen is an important gas. That gas is used in this electron transport. So oxygen plus protons plus electrons all together form as an H2O. So water formation in this electron transport is called metabolic water or respiratory water. The complex pi, last complex is it is ATPase. ATPase otherwise called as a complex pi. Actually based on the enzymes there is in four complexes are present in complex 1, complex 2, complex 3, complex 4 along with some coenzymes, but complex pi is purely only ATPase enzyme is present. Based on that protons gradient, protons are moving from high concentration to low concentration. When the protons are moving from high to low concentration, ATP. Now we can try to understand the balance sheet of ATP formation at end of this electron transport, means followed by glycolysis, Krebs cycle, link reaction and electron transport. In the glycolysis, the first of all in the glycolysis, then two ATPs are consumed. That one ATP is consumed in between glucose to glucose 6 phosphate formation. The second ATP is consumed in between fructose 6 phosphate to the fructose 1,6 biphosphate. Two ATPs are consumed, not produced. The first NADH2 production is in between 1,3 biphosphoglycerol dehyde 2 1,3 biphosphoglyceric acid. Two NADH2s are formed. Then here two ATPs are formed in between 1,3 biphosphoglyceric acid to 3-phosphoglyceric acid. Then two more ATPs are formed in between 2-phosphoenol pyruvic acid to pyruvic acid. In the glycolysis, the total amount of energy consumption is 2 ATP. The total amount of energy formation is 4 ATPs. Purely in the form of ATP, the net gain ATPs are 2. Then two NADH2s are formed. These each NADH2 can produce 2 ATP after electron transport system. In the glycolysis process, the total amount of energy production is the total amount of ATP formation after electron transport system is 8. But that amount of energy consumed to start the glycolysis is 2 ATPs. Mean that net gain ATP is purely ATP form of energy is only 2 ATPs, but after electron transport the total amount of ATP form of energies is 6 ATPs. Now we can see that Krebs cycle and link reaction, then formation of energy. The first one is in the link reaction, two NADH2s are formed because of two pyruvic acids undergoes to link reaction. Before enters the mitochondrial, there is a link reaction is there. Each pyruvic acid can produces one NADH2. The total amount of NADH2 in the link reaction is 2. In the sense, each NADH2 can produces 3 ATP. The amount of energy production in the link reaction is 6 ATPs. That also energy is produced in between pyruvic acid to that acetyl coenzyme A. Then after Krebs cycle, in the Krebs cycle, then 4 NADH2s including with the link reaction. But actually only 3 NADH2s are formed in the Krebs cycle process. That one NADH2 is formed in between isocitric acid to the oxalosuccinic acid. They also can produce, each one can produce 3 ATP, in the sense 6 ATPs are formed. Then one more NADH2 is produced in between alpha ketoglutarate acid to the succinyl coenzyme A. Then succinyl coenzyme A to the succinic acid, one GTP is produced from each one, in the sense 2 GTPs are formed in between succinyl coenzyme A to the succinic acid. Then two FADH2s are formed in between succinic acid to fumaric acid. Each FADH2 can releases 2 ATP. Each GTP is equal to 1 ATP. GTP in the sense guanosine triphosphate, FADH2 flavin adenine dinucleotide phosphate. FADH2 as well as GTP. GTP is equal to ATP. FADH2 is equal to 2 ATPs. The last NADH2 formation is in between malic acid to the oxaloacetic acid. One more NADH2 is formed. Means the total number of NADH2 including link reaction is 4. The total number of NADH2 other than link reaction is only 3 NADH2s are formed. Each NADH2 can produce 3 ATPs. In the sense, including link reaction, the NADH2's number is 4. 4 into 3. Then here 12 ATPs are formed. Only by the NADH2, 4 ATPs are released from NADH2. In the form of NADH2, 4 ATPs are released. Ultimately, the total amount of energy production is 36 ATPs after consumption of 2 ATPs. Means total amount of energy produced, actually grand total is 38 ATPs are produced, then 2 ATPs are consumed in the initially, so the net profit, the net gain ATPs are only 36. Here that NADH2 converts into ATP, FADH2 converts into ATP, GTP converts into ATP. Respiratory quotient. The respiratory quotient is defined as the ratio of carbon dioxide produced to oxygen consumed per unit time by an organism. RQ is equal to carbon dioxide by the volume of carbon dioxide by the volume of oxygen. Oxygen uptake during respiration can be measured using a respirometer. There is a small topic of respiratory quotient. Then respiratory quotient is defined as the ratio of the volume of carbon dioxide given out to the volume of oxygen absorbed in the respiration. Means the rate of carbon dioxide production by the rate of oxygen consumption. For carbohydrates, that RQ value is equal to 6 carbon dioxide by 6 oxygen is equal to 1. Means respiratory quotient for carbohydrates is 1. For lipids, the respiratory quotient is 0.70. Means the amount of carbon dioxide released out is 1 or 2 molecules. Then amount of oxygen is consumed. For fats, respiration is 145 oxygen molecules. Means the amount of oxygen consumption is more than the amount of carbon dioxide releases. Then respiratory quotient is falls down, then 1 means 0.7 respiratory quotient for lipids. It is a small topic. Then we can see the few more substances and the respiratory quotient. Malic acid produced in succulent plants. In these plants, the amount of oxygen consumption is 3. The amount of carbon dioxide emission is 0. So respiratory quotient in the succulent plants is 0 by 3, nothing but 0. Then here some of the organic acids. Malic acid in common plants, the amount of carbon dioxide emission is 4. The amount of oxygen consumption is 3. So the respiratory quotient in other than succulent plants is 1.3. Oxalic acid. Then here the amount of oxygen consumption is 1. The amount of carbon dioxide emission is 4. So the respiratory quotient of oxalic acid is 4. Tartaric acid. The oxygen consumption is 5. Carbon dioxide emission is 8. 8 by 5. So that's 1.6 respiratory quotient for tartaric acid. Then some different concept in that alcoholic fermentation. The respiratory quotient is infinity because of the amount of oxygen consumption is 0, but the amount of carbon dioxide emission is something more than 1. So then in that alcohol fermentation 2 by 0 is equal to infinity. Respiratory quotient of the alcohol fermentation is infinity.